What's up, guys? Happy New Year. We are back. And this week, we are breaking down Robert Lustig's appearance on the Andrew Huberman podcast. But first, make sure you like the video, subscribe to the channel, and leave a comment for the algorithm. A couple weeks ago, Dr. Robert Lustig appeared on the Huberman Lab podcast. Many people reached out to me asking for my opinion of his appearance. Now, I will give my personal bias. I've heard of Lustig for over a decade now. I'm pretty aware of many of the claims he's made and was already aware that he has made many claims that were not supported by data. And so that's my personal bias coming into this. I also like Andrew Huberman. People have reached out to me and said, hey, I've heard some things on his show that you said you didn't agree with. That's all true. But here's what I'll say about Andrew that I really like and value. The way I actually got on Andrew's podcast the first time was I actually called him out on Twitter for a claim he made around alcohol. I posted studies and Andrew actually reached out to me and said, hey, I wasn't aware of that research. Thank you so much for bringing it to my attention. I'll make sure that I amend my claims in the future. And then we kind of chatted back and forth, and he was like, I'd love to get you out for a podcast, and that was kind of the way the whole thing went down. I will always have a high degree of respect for anybody who can change their mind, especially with a big platform when they make a claim and then are willing to change it when they're shown actual data. That is part of scientific integrity. And people, when I was talking about some of this stuff, because I was posting some of it on Instagram, they says Andrew's responsibility to, break these claims down and, look at all this stuff. Listen, guys, it took me, I didn't even get through the full thing. I'm gonna be honest, I didn't get through the full thing. I stopped, you'll see where I stopped and why I stopped. But it took me probably 15 to 20 hours to listen to it, write down the claims, analyze the claims, get the relevant citations, to compile everything together, and then to film this video. It takes a really long time. It takes a disproportionate amount of time. And if he was going to do that, he wouldn't have time to film that podcast. For every single episode, he wouldn't have time to film it. And if he wanted somebody to do it for him, you would need individual research scientists in those particular fields, and it would cost a lot of money. What I do value, again, about Andrew is he actually has a link where you can upload papers that may disagree with something he has said or one of his guests has said. And so I'll actually include that link here. So it's where you can upload documents, and I'm actually going to upload my document for him after I film this. I think Andrew may not always get it right, but he is trying to get it right, and I can appreciate that, and I'll appreciate anybody who's willing to listen when somebody breaks down claims that have been said. And my personal bias, I obviously was on Andrew's podcast. I met him. I like him on a personal level. So there's my personal bias in that. On to the actual podcast with all that stuff out of the way. Before I start breaking down the individual claims, I want to talk about the theme that will keep popping up in this podcast. And that is Lustig gets really focused on individual biochemical pathways and mechanisms. People think when they hear a mechanism, when you hear a biochemical pathway, they think automatically outcome. And so let me give you an example of that. And Lustig actually used this. Insulin inhibits lipolysis in fat cells and increases lipogenesis. Therefore, if you're eating a high carb diet, it is going to make you fat regardless of calories. That, what we're talking about, insulin's effects on lipolysis and lipogenesis, those are mechanisms. An outcome would be if we feed people diets that produce different levels of insulin with different amounts of carbohydrates, but we hold calories equal, do we see differences in the actual loss of body fat? So an outcome is when you're measuring the thing that you're actually concerned about in humans. And when we measure that, this very thing I talk about, and I'm going to show you the paper I cite for it, there is a small statistical difference, and it favors a low-fat, high-carb diet over a low-carb, high-fat diet. Now, it's not much. It's a very inconsequential and clinically irrelevant amount, but it is more loss of body fat. So what do we care more about? Do you care more about this mechanism of insulin having this effect on lipolysis in the short term? Or do you care about, do these subjects lose more body fat? I care about the loss of body fat. A way to compare this would be like a mutual fund versus an individual stock. So outcomes are the summation of not just one mechanism or one biochemical pathway, The loss or gain of body fat as an outcome is the summation of hundreds, if not thousands, of different pathways summing together to produce that outcome. And when you focus on one pathway or even a couple, you are completely neglecting other pathways. And many times you can have one pathway get activated by something, and there's another pathway that acts in opposition that also gets activated. Aspirin is a great example of this. We know that the outcome of aspirin is it reduces coagulation, it thins blood, but it also, there's certain things that aspirin does that are like somewhat procoagulant. Do you care that it's doing this procoagulant one thing, or do you care about whether or not it actually reduces coagulation? If we look at outcomes, outcome is like a mutual fund, which a mutual fund is made up of a bunch of funds, a bunch of stocks. Somebody focusing on a mechanism would be like somebody looking at an individual stock in a mutual fund of 500 stocks and saying, ooh, you don't want to invest in this mutual fund. Look at this stock in this mutual fund. It's down by 40% this year, while neglecting to tell you that the overall mutual fund is up by 30%. Do you care about the individual stock that's down, or do you care about the mutual fund that's crushing it? You care about the mutual fund because that is the outcome. And just so you guys know, I am going to be having some notes here because there's a lot of stuff to go through and a lot of citations to remember. So if you see me look down, I apologize. I usually try to be very engaging with these, but there is a lot of stuff to cover in here, and these notes are going to help me. So all that out of the way, let's get into the claims.
Everyone thinks that obesity is about energy balance. Calories in, calories out. Therefore, two behaviors, gluttony and sloth. Therefore, if you're fat, it's your fault. Therefore, diet and exercise. Therefore, any calorie can be part of a balanced diet. Therefore, don't pick on our calories, go pick on somebody else's calories. This is actually what the food industry uses to assuage their culpability.
Now, I'm going to come back to this claim specifically, but a calorie is a unit of measurement. All calories are the same, just the way all seconds on a clock are the same, just the way that all miles per hour on a speed dial are the same. All sources of calories may not be the same because different sources of calories may have differential effects on appetite and energy expenditure, and he touched on it, which is protein, for example, causes a greater increase in the thermic effect of food and may have a greater satiating effect, same thing with fiber. But that doesn't mean that calories are all somehow different. And this quote, that if you gain too much weight, it's because you're a glutton or sloth. So I'm going to spoil it and just say that he's telling you the reason you gain weight is not because of calories, it's because of insulin, because of refined carbohydrates. So let me get this straight. They're trying to escape culpability because it's calories, and they're saying you're a glutton or sloth if you ate too many calories. And no, it's not that. How is it less gluttonous to say you ate too much sugar? You're saying the same thing. They ate too much of something. Calories in, calories out, it doesn't mean that you're a glutton or a sloth or lazy or anything like that. It just means if you gained body fat, you ate too much for your given level of energy expenditure for the day, period. And he says they use this to escape culpability. I'm not sure how that makes the food industry less culpable when food has become more palatable, way more palatable, and more energy dense. If anything, if you take a look at calories on food labels of very palatable, energy dense foods, there's a lot of culpability there because there's a lot of calories there. This is not a difficult thing, but this is just him trying to shift away and kind of minimize the importance of energy balance because it kind of screws up his whole narrative as we go.
Fructose is addictive, activates the nucleus accumbens, the reward center of the brain, in the same way that cocaine, heroin, nicotine, alcohol do. And fructose has no function in the human body. Fructose consumption's gone up 25-fold since the beginning of the last century.
I could find no evidence to really support that. This idea that it activates the same center of the brain as all these addictive drugs. So do hugs. So does skin's contact with a loved one. Does that mean you're addicted to those things? No, and this is what we call a non-equivalency fallacy. If I'm driving a car at 5 miles an hour, I'm driving a car. If I'm driving at 100 miles an hour, I'm also driving a car. But those two things are not equivalent. I've put up a couple papers here showing that there's actually no evidence for sugar addiction in humans. So he makes this case that these things are addictive. The research has not supported that. In fact, I'm actually going to review another paper today for a separate video where we actually showed that sugar and processing have less to do with how palatable something is, and actually texture made a bigger difference than processing or sugar intake. The idea of food being addictive or addictive foods, there's not a ton of evidence to support it, not specifically sugar. Now there is what's called food dependence. There are foods that are harder to not consume, but those are not just high in sugar. They tend to have a bunch of different characteristics that is basically just they're ultra processed, very tasty, typically high in sugar and fat, and they have salt and a certain amount of mouthfeel. Like there's a lot of things that go into hyperpalatability. It's not just sugar. And again, if sugar was addictive, the cheapest form of sugar is getting a bag of table sugar. Why are people not taking bags of table sugar and just going, ah?
Fruit is okay because of the fiber, and the fiber is what mitigates the absorption.
I could find no evidence that fiber inhibits the absorption of fructose. None. Additionally, the amount of fructose in 200 calories from an apple versus 200 calories from a soda is pretty close. 24 grams of fructose in an apple, 28 grams of fructose in 200 calories from a cola. Now, it's much more easy to overconsume the cola, I'll give you that, but the amounts are similar. I think he actually used to say don't eat fruit, but now he kind of says it's okay. I could be wrong on that. You know, he has to make this case that fruit is different because we look at all the beneficial effects of fruit and how many studies have shown that fruit makes you healthier. You know, he has to come up with a reason why that is. Now, I'll tell you why that is. It's because fruit has fiber, yes, but it's also that it's harder to overconsume. It has a bunch of phytonutrients and those sorts of things. But the fructose in fruit, this is one thing that I realize that a lot of people don't understand. The fructose molecule in fruit is the exact same chemically as the fructose molecule in table sugar and the fructose molecule in high fructose corn syrup. There is literally nothing different about them. If you get 20 grams of fructose from high fructose corn syrup versus 20 grams of fructose from an apple, it is the same fructose, period.
If you basically eat ultra-processed food, high in sugar, for 10 years in a row, it's going to show up in terms of your comorbidities. And ultimately, yeah, it will kill you.
If that's true, how do you explain high fructose intakes from sucrose and cereals are actually associated with decreased mortality? Some of these foods you're talking about, like these cereals, there's a study right here associated with decreased mortality. How do you explain that data? It is incongruent with your hypothesis. And when it comes to the individual sugars, when they categorize them into the different monosaccharides, fructose was no greater risk for mortality than any other sugar. Here's a personal favorite.
This whole calories a calorie just makes no sense. And it hasn't worked at any level, and there is no study that actually shows that cutting calories makes a difference. And I can show you, voluminous data that shows that virtually every weight loss study that led to, caloric restriction basically didn't work. Not for any length of time.
He threw that last little bit in there for semantics because it's the same thing the crazy anti-diet people say. As far as his statement, this is complete and utter nonsense. There are thousands of studies demonstrating that calorie restriction causes weight loss. Now, the reason that calorie restriction tends to not work long-term is the same reason that any diet tends to not work long-term. Because people do not stay on them long term. They do not adhere to them long term. And that's true for low carb, low fat, intermittent fasting. Pick your popular diet. None of them are better long term for adherence than others. But we have tons of studies. I mean, if you printed off the papers, it would literally be metric tons of studies showing that if you decrease calories, you decrease body weight. I've cited a few. That's actually so insane, it's almost not worth debunking because I just can't imagine anyone really believes that. I can't even imagine that Lustig believes it.
Insulin is the bad guy in this story. The higher your glucose goes, the more your pancreas will release insulin in order to bring that glucose down. Well, it turns out that glucose rise was not benign. That glucose rise led to endothelial dysfunction. Transient, but nonetheless endothelial dysfunction.
So he wants to say that if you have a rise in glucose, it leads to endothelial dysfunction. I can make this argument, you eat this after a meal and this happens, I can make this argument for literally any nutrient being bad for you. Let's take fats. Eating high dietary fat, especially saturated fat, can reduce flow-mediated dilation. Flow-mediated dilation reduction is a risk factor for cardiovascular disease. Okay, don't eat carbs because glucose and endothelial dysfunction, but also don't eat fat. Protein increases mTOR activation. mTOR is a complex that is dysregulated in cancer and overactive. Therefore, don't eat protein. Also, exercise in the short term increases inflammation, increases reactive oxygen species, increases your blood pressure, increases your heart rate. All those things are bad, so don't exercise. So you're left with eating nothing. Also, don't do anything, don't do any activity, because that's also bad based on this logic. What's interesting is I actually found a study where when you substituted fructose for other sugars, it actually improved HbA1c by a little bit, which is a marker of long-term blood glucose regulation. Again, he's getting focused on short-term mechanism and neglecting the long-term outcomes. Again, if we focus on the mechanism, in the short term, exercise significantly increases systemic inflammation. Systemic inflammation is associated with an increase in the risk of heart disease. What does exercise do in the long term? Oh wait, it decreases your risk of heart disease. So what do we care more about? Measuring a surrogate marker in the short term or measuring the freaking thing that we actually care about? Once again, focused on a short-term mechanism versus the actual outcome. Now, Lustig also made some claims about cancer, and I am not a cancer expert. And many of these claims were very mechanistic based, which is kind of how this whole thing went down. I don't feel comfortable really discussing those, so I reached out to a friend of mine, Dr. Joseph Zundel, who is a PhD in cancer biology, has conducted and published real cancer research, and is an expert in cancer biology. So we're going to throw it over to him to let him break down some of these claims. Joe, over to you.
The cell has to know when to grow and when to burn. It turns out that the signal for that is oxygen, because oxygen is necessary for mitochondria to be able to burn. In the absence of oxygen, the cell only knows how to grow.
Now, in addressing this claim, I can't help but point out the juxtaposition here. If oxygen is necessary for mitochondrial respiration, then why would oxygen deficiency promote growth or cell survival? Now, we know this to not be true, as oxygen drives a process called oxidative phosphorylation in the electron transport chain in the mitochondria itself. This basically supplies the cell with enough ATP to perform its various functions, generally speaking. Now, I can say here that oxygen availability throughout the body in a normal, healthy individual will change the potential of the ETC and how much ATP is generated. I can say with a strong degree of confidence that in a healthy individual, oxygen promotes metabolism of cells which are metabolically very active. Things like skeletal muscle, the brain, skin, etc. While this doesn't always translate to changes in cell growth, in general, oxygen promotes processes which sustain metabolism for cell survival. This is something we learn in basic cell biology, and it becomes more complex in diseases like cancer.
This is actually why Otto Warburg won the Nobel Prize in 1931 for the Warburg effect. He asked the question, how come cancer cells don't need oxygen to grow? And the answer is because no cell needs oxygen to grow. In fact, oxygen is the thing that prevents growth.
So Otto Warburg discovered what is now called the Warburg effect. In summary, Otto observed that cancer cells have increased glucose uptake compared to normal cells. Now, what was strange to them at the time was that cancer cells, alongside increased glucose uptake, they were making lactic acid, which is typically a product of fermentation. Now, as you know, fermentation doesn't happen under normal circumstances, especially in the presence of oxygen. Warburg and his colleagues also noted that fermentation as a result of lactic acid production was varied in different parts of a transplanted rat tumor. Now, this shows that oxygen gradients exist throughout a solid tumor, from which Warburg himself states that fermentation is not sufficient to maintain the life of cancer cells in oxygen-deficient areas of a tumor. People have a generalized view that a tumor is one type of cell, but solid tumors in particular have many layers that often function independently of each other. As such, Warburg's initial studies showed that over time, oxygen and glucose deficiency killed cancer cells, which shows that cancer cell metabolism isn't primarily driven by oxygen or glucose availability. Now fast forward to 2024. We now know that in solid tumors, hypoxic regions, which are low in oxygen, typically at the core of an advanced tumor, will secrete molecules that prompt blood vessels to grow towards the tumor. This is a process called angiogenesis, or blood vessel formation, and is a hallmark of cancer, just as the Warburg effect. Tumors do this to bring oxygen via hemoglobin into cancers because it is necessary for most cells of a tumor. Importantly, hypoxic cores are typically dead or dying parts of tumors because they don't get enough oxygen. That's why they need blood vessels. Additionally, we also know that cancer cells will use Warburg metabolism, or increased glycolysis rates, to increase citric acid cycle substrate production. The citric acid cycle can produce intermediates which can be routed into pathways that can prompt nucleic acid synthesis, things like RNA and DNA, protein synthesis, and fatty acid synthesis that can sustain cancer cell metabolism decoupled from the electron transport chain for ATP generation. So cancer cells contextually need oxygen to grow, but they are capable of changing their metabolism to be less dependent on oxygen. Cancer cells often, but not always, rely on increased glucose absorption and breakdown as a primary source of fuel.
Every fetus is oxygen deprived. Normal partial pressure of oxygen, 100 millimeters of mercury out here, right? If I checked your blood, it'd be about 100, right?
I hope so.
How about a tumor cell?
Guessing tumor cells probably, is it double?
About 44.
Wait, you just told
Partial
me.
pressure of oxygen in a tumor cell is about 44. But a fetus, what's the partial pressure in the placenta? I don't know. 6 to 31.
Tumors generally have a lower partial pressure or oxygen presence compared to surrounding healthy tissue. Lustig, I believe, is referencing a study which says that the median partial pressure in hypoxic areas of breast cancers is 44. Now, the focus of the study was to establish partial pressure as a prognostic between stages of cancer. This study also implies that altering partial pressure may be helpful for cancer treatment. Now, this last idea is where people misinterpret our meaning in that something like hyperbaric oxygen would be helpful in treating cancers. So realistically, we have a greater likelihood of changing tumor partial pressure through small molecule methods which change tumor metabolism that disrupt tumor cell ability to use oxygen, like the FDA-approved inhibitors of angiogenesis. Now, it is important to note that providing oxygen to the wrong part of a tumor could make things much worse.
Tumor cells are some of the most vascularized cells, or tumors are heavily
they're
vascularized.
trying
I mean, one way to try and kill one is to devascularize a tumor cell.
Yes, and angiogenesis inhibition, et cetera, is a big deal. Judah Folkman and all that from Harvard. Yeah.
So the excess blood to a tumor is the attempt to bring in oxygen that it's not getting.
That's right.
As opposed to delivering lots of oxygen, and that's why it's growing.
That's right.
So Andrew is correct here. We use inhibitors of angiogenesis to restrict oxygenation of tumors. This point is actually counterproductive to what Lustig is claiming, because tumors recruit blood vessels to get more oxygen because it is necessary for cancer cell growth. And yet Lustig is agreeing with the premise. I find that hilariously in support of how stupid Lustig is. Andrew is proving him wrong, and he is just digging deep in his false convictions. Now you can see here that Andrew is confused by Lustig's points regarding oxygen and cancer growth because of his multiple self-contradictions. At this point, Andrew should have asked, if oxygen wasn't so important for tumor cell growth, then why would the tumor recruit blood vessels for oxygen via angiogenesis?
Has there been any effort to treat tumors by oxygenating tumors?
Yes.
And what does that look like?
It's hyperbaric oxygen therapy. It's a thing.
Of note, Warburg and his colleagues also used a primitive form of hyperbaric oxygen therapy in some of his studies. He witnessed decreased tumor growth, which is contradictory to Lustig's point. In his studies, Warburg mentions that most parts of the tumor were affected by increased oxygenation. The oxygenation appeared to have damaged the capillaries, which likely diminished oxygen conduction into the tissue. In this, Warburg hypothesizes that it isn't the oxygenation of the tissue that kills the tumor, but the destruction of the capillaries that deliver the oxygen. We know now that capillary physiology needs a certain amount of oxygen to maintain capillary pressure. Otherwise, it will collapse. In other words, too much oxygen is bad, too little oxygen is bad. In general, currently, there is insufficient data to suggest that hyperbaric oxygen therapy will be a useful approach for cancer. It is too non-specific to cancer tissue, especially in late stages.
Thanks, Joseph. Really appreciate you breaking that stuff down. It appears that while it sounds very smart, the way he is breaking down these mechanisms, when we look at the overall body of literature and the actual outcomes, it's just not supported by the stuff he's saying.
So the glucose has to be phosphorylated, so you're going to lose an ATP in the process. So you're going to go ATP goes to ADP, and then that ADP will go to AMP, adenosine monophosphate, which will then go to IMP, inositol monophosphate, which will then go to uric acid.
Okay.
And that uric acid will be then released from the cell, circulate in the bloodstream, and hopefully go out in the kidney. Mm-hmm. In the process, that uric acid can inhibit mitochondrial function, and it can also inhibit endothelial nitric oxide synthase, which is the enzyme in your vasculature that is your endogenous blood pressure lowerer.
Right, by expanding blood vessels and capillaries.
Whoa! So this is a fallacy of if A to B happens, then B to C will happen, then C to D will happen, then D to E will happen, and so on and so forth. That's, again, very focused on a biochemical pathway. There is a lot of stuff in the body that requires ATP to be hydrolyzed to ATP, like literally any enzymatic reaction in the body, not any, but many enzymatic reactions in the body require ATP hydrolysis. But you're worried about glucose? You're not worried about the sodium-potassium pump? Because that's going to be chewing up way more ATP. You're not worried about that? Why is that not causing uric acid to rise? Focus on the mechanism versus an outcome.
Fructose will, first of all, go into the intestine. The intestine will metabolize some of that fructose through what is known as intestinal de novo lipogenesis. About 10% of that fructose will be turned into fat right in the intestine.
So de novo lipogenesis is the synthesis of fat from non-fat substrates, such as fructose or glucose. So I looked this up. Consuming fructose does not increase intestinal de novo lipogenesis in humans. There's the citation. Go look it up. He's looking at, well, in these rat studies, fructose increased the activation of this enzyme in the intestine. Let's just go measure the dang thing. Do we have studies that actually measure the dang thing we're interested in? Oh, wait, we do. In humans, wouldn't you know? And wouldn't you know it, it doesn't do that in humans. Now, the study did find that fructose increased triacylglycerides in the blood. Now, this was at a very high dose of fructose, by the way. But it's because it reduces triacylglycerol clearance, not because it's increasing triacylglycerides production.
Fructose is a driver of leaky gut. That causes inflammation at the level of the liver, which ultimately leads to systemic inflammation. One of the reasons why high sensitivity CRP is high in patients who eat ultra-processed food. And 93% of Americans today are inflamed.
Does that mean that 93% of Americans have leaky guts?
Yeah, it does.
Where is he getting this data from? This 93%? I'm unaware of it. I couldn't find it. Again, he's saying A to B, then B to C. He's doing this same thing again with these mechanisms. He's saying fructose is going to increase inflammation because it's going to cause leaky gut, which is going to cause inflammation, and so on and so forth. What if we just measure whether or not giving fructose increases inflammation? Oh, we have studies in humans, and oh, it doesn't. Not when you don't overconsume it. Not when you're not consuming too many calories. People who eat high amounts of ultra-processed food have high levels of inflammation because they consume more calories, they have higher levels of body fat, and they also tend to have a bunch of other unhealthy lifestyle behaviors, like they exercise less, they have more sedentary jobs, they're more likely to smoke, they're more likely to drink alcohol, they're more likely to have an overall unhealthy lifestyle. It's not from the fructose.
Clearly can't be the transition away from trans fats that's increased obesity.
Well, no.
So it's going to be the increase in sugar and
the increase in sugar
and these hidden sugars in foods.
Exactly.
So actually, over the last 30 years, sugar consumption has actually dropped, while obesity and diabetes has continued to climb. Here you go. And here's a really nice graph from Stefan Guillenet, kind of showing sugar intake fell off and obesity continues to rise. And in fact, the last few decades, the biggest source of added calories is actually from dietary fat, mostly oils. And people don't talk about this, but overfeeding dietary fat also can induce insulin resistance. We have plenty of studies to show that. If you gain body fat, you will become less insulin sensitive, unless you're like crazy lean and you're just getting back to a healthy body weight. Like if you're emaciated and going back to healthy body weight, that's not true. But if you're at a healthy body weight and now you gain body fat, you will become less insulin sensitive because adipose tissue secretes adipokines and all kinds of pro-inflammatory cytokines that negatively interact with insulin sensitivity. There's lots of reasons why insulin sensitivity is reduced as you gain body fat, and that's one of them.
My colleague, Dr. Efrat Monsenigo Ornan, who is the chairman of nutrition at Hebrew University, Jerusalem, actually looked at this question and showed that ultra-processed food actually inhibits growth. It inhibits cortical bone growth. It inhibits trabecular bone growth. It inhibits cancellous bone growth. It inhibits linear bone growth. It hijacks growth for cancer because it inhibits mitochondria. And so you have to then grow instead of burn.
It is important to note that the study Lustig is referencing was performed in rats. I actually couldn't find a human data counterpart to support the study. Now, upon my research of the lab in question, they only do rodent research, and they seem to publish the same mechanistic ideology through various publications, as is common in academia. The study Lustig referenced wasn't even about cancer. Lustig contradicts himself yet again in addressing the comment about processed foods hijacking cancer cells and mitochondrial inhibition. If ultra-processed food consumption inhibited mitochondrial processes and therefore growth, then why in cancers associated with ultra-processed food consumption do we see enhanced mitochondrial biogenesis and growth? So his argument makes no sense. For example, in many cancer types, we see increased expression or activity of enzymes involved in the citric acid cycle. The citric acid cycle primarily takes place in the mitochondria of cells. Contextually, cancer cells can increase the rate of enzyme substrate kinetics within the mitochondria to sustain the energetics associated with cancer cell metabolism. In fact, many cancer cells increase the number of mitochondria they have in their cells compared to normal cells to sustain their metabolic needs. Clinically, we have many FDA-approved inhibitors against mitochondrial metabolism in specific cancers to inhibit mitochondria. So his point is not well formed. Ultra-processed food consumption is associated with increased risk of mortality for a variety of reasons and does predispose someone to getting cancers of the colon, liver, pancreas, kidney, breast, ovaries, and a few others. People don't just eat ultra-processed foods and get cancer. They get other diseases like diabetes, heart disease, liver disease, and obesity first. I studied liver cancer for a bit and am actually back in the liver cancer realm as of late. Regarding liver cancer and the Western diet, we see that ultra-processed food consumption increases risks for fatty liver disease and eventually cancer, like hepatocellular carcinoma, if behaviors associated with consumption of ultra-processed foods are not changed. Added sugars are of interest here in a person's diet because if a person isn't consuming a balanced nutritional profile, then these added sugars are correlatively linked to microbial dysbiosis in the gut, altered insulin signals in the pancreas, and altered glycogen and fatty acid metabolism in the liver. We know that fatty acid composition, particularly from saturated fats of ultra-processed foods, also play an important role, especially regarding caloric intake, low fiber content, and low vitamin content. Realistically, all of these factors come into play and sugar alone cannot be blamed. Regarding research on sugar, of course, we need to isolate these variables to determine their contributions to diseases.
How much of that weight loss effect do you think is a calories in versus calories out effect? And how much of it do you think is the elimination or near elimination of this fructose or this glucose-fructose combination?
It's nothing to do with the calories. It has everything to do with the insulin.
Categorically untrue. Let's look at the actual, wait for it, you guys are ready for it, you know what's coming, human randomized control trials! In a meta-analysis from 2017, they looked at human randomized control trials where calories and protein were equated between different diet types, which is important because protein is thermogenic, as we've discussed, and varied the amount of carbohydrates and fats. So anywhere from low carb, high fat to low fat, high carb. And they looked at fat loss, like actually looked at fat loss. You know, the thing we actually care about. And what did they find? Oh, I'm so happy you asked. They found actually a small but statistically significant advantage for low-fat diets. It was basically clinically meaningless. It was an extra 16 grams of body fat lost per day on low-fat diets compared to low-carb diets. And what's important is these studies were very well controlled. They supplied all the foods to the participants, and some of them were in metabolic wards. These are the highest, most controlled, most rigorous studies you can do for dietary interventions. If it had nothing to do with calories and everything to do with insulin, we should see a linear effect that as carbohydrates go up, fat loss goes down. And we do not see that. And in fact, there was one study that took this even more granularly, where they took obese men and either fed them a high-carb, low-fat diet, and by high-carb I mean over 300 grams of carbs a day and under 20 grams of fat, or they were switched to a low-carb, high-fat diet. I think it was like less than 20 grams of carbs a day. I can't remember the fat amounts. And they looked at actual loss of body fat and insulin levels. Well, the low-carb diet had much lower insulin levels over a 24-hour period. And they actually lost a little bit less fat. Again, mechanism versus outcome. If insulin was so critically important for fat loss, why do we not see it show up in the human randomized control trials?
In addition, branched-chain amino acids make insulin go up as well. Leucine, isoleucine, valine, which is in corn-fed beef, chicken, and fish, processed food. Get rid of the branched-chain amino acids. Okay, so eating fish is a good place to be. Even eating a steak is okay if it's a pasture-fed steak. You consume those excess branched-chain amino acids, they're going to go to the liver, they're going to be deamidated, like we talked about earlier, and they're going to end up as branched-chain organic acids. They're going to flood the mitochondria. The mitochondria are not going to be able to deal with the volume, and so they're going to divert the excess and turn that into fat.
So, as someone who did their PhD in protein metabolism, specifically focusing on the branched-chain amino acids, BCAAs, the idea that when they're deaminated, they're going to turn to fat is insanity. Is there a pathway where they could eventually become body fat? Sure. And what do we see in the studies where people are overfed high-protein diets? They don't gain a bunch of body fat. Mostly because protein's satiating. But they don't gain a bunch of body fat. That doesn't happen. And the BCAAs, like he likes mechanistic studies? Well, here's one. In lab rodents, when they knock out the enzyme that causes the catabolism of BCAAs, those rats don't get fat. They stay lean. And they have very, very, very high circulating levels of BCAA. If you like mechanism studies, there you go. Now, he says, get rid of the high BCAA foods and eat fish instead, even though like earlier in the thing, him and Andrew were talking about having a nice porterhouse steak. Okay. The BCAA content of fish is very similar to any other animal protein. The BCAA content in fish is about 22 to 23% of the total protein content. In beef, it's around 26 to 27%. So you're telling me that a 3 to 4% difference is going to make you fat. Like we're talking about milligrams of BCAAs here. This doesn't make sense. And then he's saying eat pasture-fed instead of grain-fed because of BCAAs. I looked for hours. I could find zero evidence that the BCAA content of pasture-fed versus corn-fed was any different between those two. And honestly, it doesn't make sense. What tends to happen with pasture-fed is they have less levels of fat, and it tends to be more like monounsaturated fat compared to a higher level of saturated fat in grain-fed. But why would it change the amino acid composition? These are ruminants. They're ruminating this stuff. The amino acid composition is going to be similar. Like the amino acid composition of the muscle tissue is created by the DNA of those cows. So what you're saying, Lustig, is somehow pasture-fed is going to change the cow's biology to get it to produce a different skeletal muscle amino acid composition. The amino acid composition of the skeletal muscle of any animal is encoded in their DNA, okay? And when it's stimulated, when protein synthesis is stimulated, it prints, best way to describe it, mRNA. That mRNA is then translated into a protein. And that protein can go to make, again, skeletal muscle. If you're going to change the amino acid composition of the muscle of cows, you're talking about changing their DNA. It's not going to matter what they're eating. Like, this is actually just crazy talk. Now, dietary fats are different. You can change the dietary fatty acid composition of an animal based on what you eat. I've seen no evidence that you can change the amino acid composition of the muscle. BCAAs are rich in all animal sources of protein. And oh, by the way, branched chain amino acids are one of the biggest, most important amino acids for stimulating muscle protein synthesis for healthy, strong muscle tissue. Additionally, guess what? Animal sources of protein raise insulin. He says you should have a porterhouse, even though then he goes and says, pasture fed apparently is going to do something. But don't eat carbohydrates or don't raise insulin. But there are studies showing that animal sources of protein raise insulin to a similar amount as carbohydrates. And yet, what happens when people eat high protein diets? Oh, they get better fat loss. Outcomes versus mechanisms, I guess.
For primary prevention, the mean increase in lifespan for being on a statin is 4 days.
I'm not sure where he got this from, but here's a meta-analysis of 150,000 people showing a 28% reduction in the risk of mortality and a 23% decrease in cardiovascular disease events. If that only translates to 4 days, okay, but seems like it'd be more than that.
So you're looking at people 65 to 90, and it turned out that the LDL level correlated with longevity. The higher the LDL, the longer they lived when you took out all the people who had problems. So LDL is not really the problem. And the reason is because there are two LDLs. There's one called large buoyant, and there's one called small dense. Turns out dietary fat raises your large buoyant. Your large buoyant is irrelevant.
Here we go. This is a big one the low-carb community like to drive home. They say, well, small, dense LDL, that's what's really problematic because it can penetrate the endothelium easier and stick in there. Yes, small, dense LDL particles can penetrate the endothelium more easily than large LDL particles. But large LDL particles still penetrate the endothelium. And large LDL particles carry more cholesterol. So when you look at the total amount of cholesterol that gets deposited in the endothelium from either small dense or large fluffy, it actually ends up being the same. And here's a study showing that it's basically the same. When he's talking about LDL being associated with longevity, he is cherry picking that research. It's associated with longevity in the elderly. Here's why. When you get diseases like cancer or basically any wasting disease, it lowers your LDL because you're experiencing wasting. People with better nutritional status who don't die will have higher levels of LDL because they're able to maintain their body weight better. This is what we call reverse causality. LDL isn't causing people to live longer. People who live longer are having higher LDL. But again, there's Mendelian randomized control trials, which are the gold standard for proving causality when it comes to like lifetime exposure risks like LDL cholesterol. The association with LDL and mortality still held in elderly in the Mendelian randomized control trials. So when you're actually controlling the variables that matter and not just looking observationally, LDL is still associated with increased mortality even in the elderly.
How many pounds or kilos of subcutaneous fat do you have to gain before you become metabolically ill?
I have no idea.
About 10 kilos, about 22 pounds.
This is a blanket statement not supported by data. I mean, if you gain 10 kilos of body fat, will you be more metabolically unhealthy? Well, probably for most people, but it depends on your starting point. What if you're like emaciated? Are you going to become more metabolically unhealthy from gaining 10 kilos of body fat? I don't know. Like, this is such a blanket statement. It's completely unhelpful.
The question is, what made the visceral fat in the first place? Was it calories? No. It's cortisol.
This is completely incorrect. Yeah, elevated levels of cortisol are associated with increased visceral fat, but saying calories don't have anything to do with visceral fat is completely wrong. Like, completely wrong. Numerous studies have demonstrated that overfeeding calories increases visceral fat and liver fat. So here are several citations. Overfeeding by 760 calories a day increases visceral fat and liver fat in non-obese people. Overfeeding calories by 40% above maintenance increases visceral fat. Overfeeding 40% calories increases visceral fats even at varying levels of protein. Shall I go on? Yes, I will. You can argue that overfeeding alone increases cortisol. So he's saying, well, it's not the calories, it's the cortisol. But these two things go together. because here's a study showing overfeeding increases cortisol. And we know overfeeding increases visceral fat and liver fat. These are what we call confounding variables. You cannot just pull out the cortisol and say that's causing it. Maybe, I think it's probably a passive player, that it's going up as calories go up and you're storing more fat around your liver and viscera.
When you have fat in your liver, it causes metabolic dysfunction right away. And the question is, where did that fat come from? That came from alcohol or sugar. So alcohol and sugar, most metabolically egregious because it affects the liver directly.
Listen, cutting alcohol and sugars can reduce liver fat, but here's where it gets really disingenuous. He talks about fructose and liver fat because of de novo lipogenesis. He talks about cutting sugar and liver fat. He did not talk about the nutrient, other than alcohol, that increases liver fat more than any other, and that is saturated fat. People freak out about fructose and liver fat in a head-to-head study comparing overfeeding fructose to overfeeding saturated fat, keeping calories the same. Saturated fat increased liver fat 70% more than fructose in a straight up comparison. Why was there no mention of saturated fat? Again, it's because Lustig kind of stays away from saying anything bad about saturated fat. He never outright says it, but he's kind of dog whistling it with his comments about LDL. But yeah, why is there no mention of saturated fat? The fact of the matter is, overfeeding anything can increase liver fat. We have numerous studies to show this. Overfeeding polyunsaturated fats or saturated fat increases liver fat, but saturated fat increases liver fat far more than polyunsaturated fats. Also, sugar and fructose do not increase liver fat or body fat compared to other sources of carbohydrates when calories are equated between diets. There are 2 large review papers going through several randomized control trials that show this. Again, if you do not overfeed fructose or sugar, it will not increase liver fat. So there was even one randomized control trial where they had people eat 150 grams of fructose per day. Soda is about 40 grams of carbohydrate per 12-ounce can of soda, about 55% fructose in high fructose corn syrup. So 55% of 40 is 22 grams. So you would need to drink approximately 7 sodas per day to get 150 grams of fructose from high fructose corn syrup. Just to put it in context. These folks fed 150 grams of fructose per day, and by the way, they didn't tell them to limit calories. They found feeding that much fructose did not increase liver fat or body fat, and these folks did not increase caloric intake. But I thought fructose was addictive. The critics of this study will say, well, it was only 8 weeks long. 8 weeks is plenty of time to show changes in liver fat. In fact, here's a study where it was only 2 weeks long and they showed an increase in liver fat. You do not need a long time to show differences in liver fat. And just getting back to the whole insulin thing, both reduced carbohydrate and reduced fat diets will reduce liver fat. If you reduce calories, it will reduce liver fat. But actually, the low fat diets reduce liver fat more than the low carb diets when calories were equated. The reason he gets so hung up on fructose is because there is a chemical pathway, a biochemical pathway, a mechanism of fructose increasing de novo lipogenesis. And in lab rats fed crazy high doses of fructose, they show increases in de novo lipogenesis. But in the human randomized control trials, even when they give high doses of fructose, it doesn't make a difference if calories are equated between diets. If you overfeed fructose, yes, it will increase liver fat. But if you overfeed saturated fat, it'll increase it way more. I'm not saying high fructose corn syrup does not contribute to liver fat, but I'm saying it does not appear to uniquely contribute to liver fat outside of the calories it contains.
That's one of the reasons why intermittent fasting works, is because it gives your liver a chance to basically offload what it's already stored. That's one of the things that intermittent fasting will buy you, is a little less liver fat.
It is true that intermittent fasting can reduce liver fat, but it does not reduce liver fat more than normal caloric restriction in studies where calories are equated between the two. And this is the last claim I'm going to address because I got this far and I honestly had to stop.
When people go on diet sweeteners, what are they doing? Are they really reducing the fat? And the answer is no, because of the insulin. Turns out there's still an insulin response.
So he's saying there's an insulin response to artificial sweeteners. This gets parroted so much on social media and people accept it as true, and it is absolutely 100% false and fabricated nonsense. Here are numerous studies showing that non-nutritive sweeteners have no effect on insulin or glycemia, including meta-analyses. And we have numerous randomized control trials that demonstrate use of non-nutritive sweeteners decreases body weight and does not increase insulin or affect glycemia. To support this, he pulls out a study.
It's a very famous study done in Copenhagen. 100 normal individuals, 25 in 4 different groups. One group, 1 liter of sugared soda per day for 6 months. One group, 1 liter of diet soda per day. One group, 1 liter of milk per day. And finally, one final group, 1 liter of water per day for 6 months. The 1 liter of soda per day in 6 months gained 10 kilos. The 1 liter of water per day lost 2 kilos. 1 liter of milk per day, full fat milk.
Yeah, they like their full fat milk.
No change. And finally, the kicker to the whole thing, diet soda. They gained 2 kilos.
This seemed weird to me. 10 kilos of body weight gain is a massive amount in 6 months, even for something like this. Something I just felt like, I'm going to go to this study and it's probably going to be 5 or 6 kilos or something like that. It just felt off. And the idea that the diet soda was going to increase body weight, that felt off, because in the human randomized control trials, where they have people use diet soda to replace regular soda, people lose weight. And usually when they compare diet soda to water, diet soda usually does slightly better in weight loss trials. I don't know how to say this any more graciously than I'm going to. The results he claimed in the study are not even in the same universe as what the study actually showed. I had to do some math to kind of normalize the values because they were doing a lot of it as percentages of starting weights. So first off, it was using semi-skim milk, not whole milk, and had 47 subjects, not 100. But not a big deal. There were no differences in body fat between any of the groups at the end of the study. Where's the 10 kilos come from? Well, let me just go through the rest of the results before I get to my reaction. So there were no differences in body fat by the end of the study. Additionally, the researchers concluded, and I quote, Diet Cola reduced systolic blood pressure by 10 to 15% compared with regular cola. Otherwise, Diet Cola had effects similar to those of water. And in fact, when we look at the raw data, here is the raw data. The soda drinkers didn't gain 10 kilos. They gained 1.25 kilos, 1.25. They gained 1.07 kilos of body fat. The milk group gained 1.29 kilos of weight and 0.5 kilos of fat. The water group gained 0.58 kilograms of weight and 0.18 kilograms of body fat. Now, these aren't statistically significant. The diet soda group gained 0.11 kilos of body weight and lost 0.18 kilos of body fat. So at that point, I honestly stopped because as I have consistently shown, when he made claims, the claims that were supported by data were cherry-picked data that did not address the human randomized control trials. And this particular case, I don't know how to call it anything else other than egregious in terms of the study. And yes, I know that was the study, 100%. Because I saw him talk about it in a blog before, and it took me a while to hunt it down, but it was the study out of Copenhagen with the exact design that he talked about, and it's not even close. So there's only really 2 possibilities from that. The first possibility is he never read the study, he heard about the results from somebody he trusted and never bothered to verify it. The second option is he did in fact read it and made it up. I think the first is much more likely, but neither of those are good. In grad school, this was beaten into my soul. If you make a claim, you have to support that claim with direct evidence of what you are claiming. That was a complete butchery of that particular study. And so I stopped at that point. Am I saying that everything he said was bad? No, he said some good things. He said fiber was good. I'm down with that. He said kind of in roundabout ways that people eat too much processed food, which they do, and they should try to limit that. But we have to get away from this fear-mongering and demonization of individual nutrients. Fructose is not the problem. Carbohydrates are not the problem. Fat is not the problem. The problem is an obesogenic food environment with cheap, highly accessible, hyper-palatable foods that do not have a great effect on satiety. That is a big part of the problem, and also the fact that we're very sedentary now. Those are the problems, but those aren't sexy. Those mechanisms aren't sexy. And he even alluded to this. People are very uncomfortable with the idea of personal accountability. And for whatever reason, you can tell somebody, well, you ate too much sugar. Well, it's because the evil food industry. But you tell them they ate too much calories. Ooh, that smarts, that stings. I don't think this is on Andrew Huberman at all. I think that as a professional, I believe Lustig is an MD, not a PhD. I believe. I could be wrong. But he has conducted his own research trial before. Now, it was a trial that is the one trial that stands in isolation where I think they took kids and took them from a diet that was higher in fructose and switched it to a lower in fructose diet, and they saw changes in liver fat. No control group, never been replicated. Am I saying he fabricated it? No, I'm not saying that at all. I'm just saying when we look at results from research studies, we make decisions and we make claims based on the consensus of the data, not single studies. I am not doing this to generate hate against Lustig. I don't know him personally. I'm sure he's a perfectly nice guy. But we cannot make excuses for this. We have to hold, especially scientists and people with advanced degrees, have to be held to an even more rigorous standard. And I know it's very difficult to sift out, well, but he said a lot of things that sounded very true and sounded very smart. I know he did. And I used to get roped up in the whole thing too before I learned how to read and interpret research. And after I learned how to read and interpret research, it became much more clear to me who was actually trying to push an agenda versus who was sticking to the evidence. What I'll say is if you haven't signed up to Reps yet, my monthly research review, that is a great way to learn how to interpret research and understand what the important things are in research and how to separate the BS from the legit stuff. But this was a very, very hard podcast for me to watch. And quite frankly, I feel bad for Andrew because I do know he likes to get different perspectives on the show, and that's great. And I'm all for different perspectives, but not when people are citing studies and making claims around them that are just absolutely and utterly false. So I hope maybe Lustig will see this. Maybe he'll amend some of his claims and do better in the future, which I sincerely hope so, because he does have a big platform. He does get to talk to a lot of people, and he has an impact. And I'm not saying everything he does is negative, nothing like that. But when it came to the claims on this particular podcast that I covered, they were at best only addressing a small portion of the evidence, and at worst, they were just outright false. All right, guys, hope you liked the video. If you stuck around this long, great job, and I'll catch you guys next week.